Hello, ladies and gentlemen. So we owe a great deal of gratitude to the mouse. It has contributed for decades to life technologies in general and to many aspects of biology. But there's actually one area where the mouse has outlived its usefulness and deserves a quiet retirement. That's the area of testing for drug efficacy. That actually doesn't work. But the question is, what's the alternative? And in the next 20 minutes or so, I hope to show you that. Every year, millions of mice are farmed for science. And finding a drug that cures a mouse is actually very exciting. We can do this relatively fast. It actually does no harm to humans. It's not so expensive. But the reality is that it doesn't work. Still, every year, 15 million people die from just the top 10 diseases. We spend over $8 trillion on healthcare. We make huge advances in science, but in phase 3, in the most critical clinical trials, still half of the trials fail just for lack of efficacy. That's actually a pretty big number. In phase 2, we actually mostly fail on efficacy too, and the question is why? Most of these drugs actually made it safely through mice models and show very promising results. So for all the mice has given us in science, the reality is that mice are not men, and not women, and not children. And they can teach us a lot about the biology through principles of evolutionary conservation, but actually they can teach us very little about the human disease. The models in mice are actually very artificial. It's not the same disease as the human disease. It's not also the same biology. Take cancer, for example. So in mice, the tumors are actually very small, the size of a grape, but in humans, they're actually closer to the size of a grapefruit. A tumor in mice evolves over a period of several weeks, but in humans, it evolves over a period of decades. The immune system, the immune response in a mouse, the immune response to the tumor is acute, while in humans, it's chronic. Plus, the number of targets we can explore in mice is very limited. Not many genes exist in a one-to-one relationship between a human and a mouse, and often they don't display the same biology. Let me show you this one example that I picked on the LILRB proteins. These are myeloid regulators that started to emerge now in the pipeline of several pharma companies. In humans, we actually have 5 different family members, but in mice, we have 2. So how can we actually explore this family and test their usefulness to cancer in mice models? Would you think that maybe we can ignore the mouse? Actually, the answer is no. What we can do is humanize the mouse. That's actually pretty cool. You can eradicate the mouse immune system. You can transplant a human immune system. Now you have all these human cells running around in a mouse body trying to figure out what's going on. We then implant a human cancer under the skin of the mouse. And what do you get? Can we actually expect to make a reasonable prediction from such a model? Actually, that doesn't work. And this is the proof that it doesn't work. This figure on the right was taken from a paper of Chen and Melman that was published in 2017. That shows all the clinical trials only in immuno-oncology running combinations. And these are ongoing trials in 2016. There are about 800. Every dot is a trial divided to the different tumor types. How many more trials do you think we got since today in 2019? The number is actually 1,700. There are 1,700 ongoing immuno-oncology trials. That's a pretty big number. Actually, if you take a rough estimate of 50 patients per trial, that's 85,000 patients in those 1,700 trials. And in immuno-oncology, the rough cost per patient is about $1 million. So the cost of this experiment is $85 billion. That's pretty amazing, isn't it? So it's based on what? It's based on a scientific hypothesis, on intuition, on hope. It's certainly not based on data. And you don't have to take my word for it. Here's a quote from one of the CEO of a big pharma company, it's definitely not based on data. This is a trial and error in a clinical setting. And the thing is, most of these trials will fail. A few of the combinations will succeed and will emerge to be the standard of care, but most of it will fail. And what have we learned from this huge exercise? Most of this data, and we're generating tons of data in clinical trials, will never see the light of day. And we won't be able to learn something new about the disease or the drugs that failed while testing on patients. There has to be a better way to do this, a more humane way. And actually, we think that there are 4 key aspects that you need to do this. I think you heard some of those today through the conference. But the first is the data. We need access to this data, and that's not trivial. You need to be sensitive to what pharma needs to safely share their data from clinical trials. You need to have a technology that leverages what they collect in clinical trials. It's not enough to just have cutting-edge technology. You need to use what is generated in the clinic. Then the data and the technology have to come together in a model to deliver biological insights that are useful. And finally, and that's like the most important part of all, I think I also heard it in some of the talks today, you need to be able to trust the outcome. And for that, you need a very stable and solid process. You need to trust it enough so that people who make decisions, that one person who sits in a pharma company and at the end has to make a decision that's going to cost his company hundreds of millions of dollars, he needs to be able to trust that result. That's just the same way he can trust seeing how a drug cures a mouse. And that's really not trivial. Currently, it requires a very strong team of biologists, computer scientists, data scientists, working together hand in hand, ensuring the process is transparent, the biology is rational, and very, very solid. So it will come as no surprise to you that we at CytoReason think we have all these 4 aspects covered. So I'm going to tell you a little bit about us. We are a fairly young company. We were born about 3 years ago. Our science is founded on over a decade of research at Stanford and at the Technion. We build disease models that help drive decisions in pharma. We have over 40 employees. Most of them have PhDs in immunology, in data sciences, in computer sciences, and bioinformatics. We're revenue funded from inception, from day one. We believe you need to bring value quickly. And we collaborate with the leading pharmas in the industry. So this is essentially our approach. It actually has 2 different aspects. One, we want to understand what cells are doing. Biology is driven by cells, but actually, if you think about it, what we measure is genes and proteins. It's very difficult to measure cells. So we developed a technology that can take gene expression data, simple gene expression data from biopsy that is being collected in clinical trials, and from that, we estimate cell proportions, because the cellular data gets lost in the process of generating gene expression data. So everything we do is centered around the cells. We need to understand the cells. The second component is we need to build all this information into a model that keeps learning. We can't look at one experiment as if it's the only experiment in the world. We want to learn from all the history. And what are we learning? We're learning the relationships between the cells and all the other omics and clinical data that are being collected. What kind of data do we have? So we collect data from clinical trials, from human biopsies. It can come from public domain, but it can also come from our collaborator, which is actually clinical data that is typically not accessible. We take all the data types, gene expression, genomics, proteomics, and so on, all the omics data types. Our models are primarily molecular. And gene expression data is the basic unit because that allows us to estimate cell proportions. Here I'm showing just for immune disorders the kind of data, the numbers we have, over 20,000 clinical samples from blood, from tissue, from normal disease and treated patients. Few words about our technologies. So our technologies have 6 building blocks. The first I briefly mentioned. We rebuild cellular information from data, and we look at cells. This is the foundational layer. We then have methods to associate between all the different data types that are being collected in the clinic. We have NLPs that actually go through the literature that are specifically trained to the immune system and provide the directionality in the model. We also have all sorts of tools that we use to query the model to infer cell-specific information that gives insights about disease models. And of course, the most important layer is statistical learning. We want to keep learning on each disease as we add more and more data. Just briefly about the model. What is the model? We have a model for each tissue and each disease. The immune system, which is the system that we're trying to primarily monitor during diseases, it behaves very differently in different tissues and different conditions. So each tissue and disease has its own model, and we're learning the associations around cells, how a cell is linked to a specific gene and how that association changes between normal, disease, and different treatments. So this was briefly about us. I want to talk a little bit about the science and to show you what kind of value we deliver, both with cell-level technology, but then also with our models. So this is the first case study, is a work we published in the journal Gut. The reference is at the bottom. It's difficult to read, but I'm happy to share it later. Here we took 3 different publicly available studies. Each had, so these are clinical trials, each had about 20 patients, so a total of 60 patients. This is an inflammatory bowel disease. Patients were treated with anti-TNF, and all the biopsies were taken pre-treatment. And patients are categorized to responders and non-responders. So this is a classical pharma question, biomarkers of drug response. And this is the conventional analysis. This is what most people do. You have 2 groups, and you look for genes that are elevated in one group versus the other. That data is actually gene expression data. Each dot is a gene. All the dots you see are significant genes differentiating responders from non-responders per cohort. Okay, so the y-axis shows how significant it is, or is more significant. The x-axis shows the effect size. And what most people do is they look at the most significant genes, like the OSM that is labeled, occurring in at least 2 of 3 studies. That's a potential biomarker. But actually, there are hundreds of genes. How do we know this is a good biomarker? So as I told you, we at CytoReason go back to the cells. There's no cell information here. We rebuild the cellular information from the data, and we ask which cells are different between the 2 conditions. And actually, that looks like that. So on the left, you see the exact same plot I showed in gene space, but in cell space. First, you can see it's much cleaner. That plot actually integrates all 3 studies. You see this at the top, macrophages, and then plasma cells. These are the only 2 significant cells differentiating between the 2 populations. So first, it's much easier to understand, right? There's less clutter. You can start thinking about the biology. Okay, why macrophage and plasma cells? They provide resistance to anti-TNF. And it's also easier to test, because for many cell types, we actually have assays for testing, which is exactly what we did. We validated this study on 52 patients in 2 different hospitals. And as you can see in the figure, non-responders have higher levels of plasma cells, for instance, and the accuracy of that prediction is 82%. So that's pretty good. But let me go back to the cells for a second. If patients that do not respond to the drug have more plasma cells than monocytes, of course, you will see many more of those genes. But you need to account for the effect. The genes are there just because the cells are there. Cells are bringing the genes with them. This is exactly the case of OSM. OSM is explained by changes in macrophages. Okay, so we developed a method to ask, okay, so we have changes in cells. Now let's see which genes are different, given that I know the cellular composition of the samples. We call this adjustment. This is the first step. Going through adjustment, we can identify genes that are truly associated with differences between responders and non-responders. In this case, we saw that many of those genes are being regulated by a protein called TREM1. TREM1 itself was not different in tissue, but it was different in blood. And we validated that on 22 patients, and indeed it separates responders from non-responders at baseline before treatment very well, with an accuracy of 94%. Okay, so that was an example of how cell-level technology can bring you a lot of value on top of what you have in the gene space. Now let me share with you in the few minutes that I have left how the model can be useful in the case of atopic dermatitis. So this is a skin disorder. And what I'm going to do here, so we took all the studies where we have normal and disease data from clinical trials, untreated disease data, untreated lesions. And what we do is we basically compare the normal to the disease states and look for cells that separate the two conditions. Then we map all the other omics data we have to the cells, okay? Just as I showed before in the model. Now, the first question, the column on the left, is which cell types are different between disease and normal? And the top cell type that comes up is a dendritic cell, and the second cell type is a Th2 cell, and there are many immune cells. Of course, all the immune cells come when you have inflammation. We also see keratinocytes. So our algorithm is tailored to a specific tissue. We can then use our NLP to go and ask which cell types are associated with this disease in the literature. And that's the second column. You see the highest scoring cells are Th2, keratinocytes, and eosinophils. Of course, this is known to be a Th2 disease. And then we can continue that way to build more and more layers on top of the models. And now we can start asking questions like, okay, let's see which genes are associated in atopic dermatitis with Th2, dendritic cell, which is the top scoring cells, and let's say keratinocyte, which is the skin cell. And that network looks like that. So the gray circles are the cells, the red are the genes, and the blue lines are actually the lines we derived from the gene expression data. So there is an association between a gene and a cell. That means that association is specific to disease condition. It doesn't exist in normal. And now we can also do all kinds of statistics and ask which processes are associated with these genes and the cells. So the bigger circles show genes associated with a specific process. In this case, it's cell proliferation. Of course, it's associated with the keratinocyte. That's the phenotype of the disease. The skin cells start to proliferate much faster. We also have a cluster of immune signaling that and we can identify between dendritic cells and keratinocytes, and that's how we can query the model. This was constructed, by the way, from over 100 patients from multiple studies. Now let me show you how we link the treatment to that. We took a clinical trial that was published. It's available in the public domain. This is work by Emma Gutmann and a group at Mount Sinai. Here they tested the antibody dupilumab, which inhibits IL-4 receptor. That's an approved drug for this disease. And they divided the data to responders and non-responders to the drug, and they also have a placebo group. Each line here is a patient, so it's not a very big study. And what I'm showing here is actually the Th2 cell contribution. They, again, did not measure cells. They measured only the gene expression. We infer the cells from the data. And the cell type that changes the most is Th2 in responders, okay? So you can see that responders have high levels of Th2 before treatment, and then those cells go down at week 4 and stay low at week 16. That doesn't happen in the non-responder. It also doesn't happen in placebo. Now, this is a small trial. You see it's a small number of patients. Typically, you don't have the statistical power for discovery. But we can leverage the robustness of the disease model. We already know that Th2 cells are important cells in the disease. How do we know it? Because Th2 went up from normal to disease, and now they go down with treatment in the responder. So these are the patterns we're looking for. And we looked at Th2 cells. We can also look at which genes are associated, how the associations have changed with the treatment, and that's actually how we get to a mechanism of action for the drug in humans. Once we crystallize that mechanism of action, we can go through all our disease model and say, which other disease model has this mechanism at the core? And that's how we can deliver value back by saying, your drug actually can work in a different indication. Then we can also look in the non-responders, and although the sample size is small, we can suggest mechanisms of resistance, and that can trigger novel targets or combinations, but it can be potentially tested with dupilumab. Of course, we can also generate biomarkers by comparing responder and non-responder, and this is how we can bring value very quickly with just a very small clinical trial data set. Okay, this is my last slide. So I hope I convinced you that it's time for a mice vacation. We've been learning on the wrong problem. You can't learn on the mouse and solve on the human. It doesn't work. We have to learn in humans to solve in humans. And what CytoReason does is provide a platform for learning on the human condition and suggesting how to solve it, and that will increase our ability to learn even more. We deliver value fast on the areas that are most painful to pharma: novel targets, indications, biomarkers, and combinations. And we hope that our method will demonstrate a much better approach to get to clinical success faster. Thank you very much.